There are currently 9 registered products to use to combat Varroa in Hawaii, with 6 being organic and 3 being conventional pesticides. Following the label for these products is very important, as some cannot be used when there are high numbers of brood, and some are not safe to use when there is honey in the hive. Keep in mind, organic does not mean safe for you or your bees. There is evidence from the mainland that Varroa is now resistant to the active ingredients in conventional treatments, though there is no evidence of resistance among the Varroa in Hawaii. To be safe, an integrated approach to control is the beekeeper's best bet. Using a screened bottom board can reduce Varroa populations by as much as 14% in the colony, and selectively removing drone brood after they have been capped is also effective at removing a whole cohort of mites, since they prefer drones. The most significant advancement, however, comes in the realm of bee breeding. Natural and artificial breeding programs have selected for bee behaviors that enhance Varroa detection and removal. One group in Ohio is even breeding bees to bite Varroa and have found 45% biting rates in their bees. So if you consistently have Varroa issues, try getting your hands on a hygienic queen and requeening the colony. The Apiary Inspectors of America recommend treating bees for Varroa if mite counts exceed 3%, or 9 mites from a half-cup scoop using the alcohol shake method. See the supplemental information for this lecture for a document summarizing the various Varroa treatment options in Hawaii. Varroa is known to transmit a number of viruses, with one of the more important being deformed wing virus. Different strains of this virus are present without Varroa, so obviously are transmitted in other ways. But research in Hawaii from 2012 explains why Varroa is so bad. First, when the mite is absent, deformed wing virus is less common. This is highlighted in the blue box, where the amount of red shows how many of the colonies sampled tested positive for deformed wing virus. Even though fewer colonies had deformed wing virus, the diversity of virus was really high, shown in the purple box. Honeybees weren't dying in these areas, so these are just background viral loads. But once Varroa was introduced, the overall amount of virus, shown in the graph on top, increased significantly on Big Island and Oahu versus the levels seen on the other islands and the Big Island before Varroa became widely established. Simultaneously, the number of affected colonies increased to 100%, and viral diversity declined, such that one virulent strain dominates in these areas today. There is nothing we can do to treat the virus, but this is another example of why mite control is so important to overall colony health. Unlike Varroa, which are visible with the naked eye, tracheal mites live inside the respiratory system of adult honeybees, meaning they are very tiny. The likelihood of detecting tracheal mites is highest when colonies are at their lowest populations in the fall and winter. Symptoms aren't easy to detect, and positive diagnosis can only be made with microscopic examinations of the dissected respiratory system. The economic impact of tracheal mites isn't clear, though when over 30% of bees are parasitized in a colony, honey production may be reduced, and the likelihood of winter survival decreases in temperate climates. Tracheal mites are present in Hawaii, though their relative contribution to colony stress is not well understood, and this is likely a background pest. However, if beekeepers can conclusively determine they have tracheal mites at economically significant levels, menthol is the only material currently registered with the EPA to control these mites. Unfortunately, this product is not licensed for use in Hawaii. Tropolaelaps are considered an imminent threat to beekeeping in the United States, though for now the distribution of this mite is still restricted to Southeast Asia. These mites are much smaller than Varroa and behave differently. If dislodged, they can actually be observed walking on the comb, whereas Varroa will almost always be observed on the honeybee brood or adults. Tropolaelaps reproduce in sealed brood cells like Varroa, but spend almost no time on adult bees. They reproduce faster than Varroa, produce more offspring, and are shown to transmit viruses to European honeybees. Consistent monitoring of colonies is critical to swift eradication in the event this mite shows up in Hawaii. In a recent survey, I asked beekeepers in the state what they thought the greatest threat was to their colonies, and 60% said small hive beetles. This pollen-feeding pest is native to sub-Saharan Africa and ubiquitous in Hawaiian hives, but is not a primary pest, meaning that if a colony dies, it's because of something else. The beetles only showed up because the colony was weak to begin with. Healthy, full-sized colonies do not succumb to small hive beetle infestations. Their life cycle is relatively straightforward. Adult beetles locate colonies by smell and fly in. They hide in cracks inside the colony to avoid harassment from the bees and lay their eggs. It is the larval stage of this pest that is the most economically damaging. The larvae feed on honey, pollen, and brood, and their feces contains a yeast that causes fermentation in hive products, making them unfit for human consumption. When this happens, it's known as a slime out. Just like honeybees, the beetles have a pupal stage. The larvae will wander to the hive entrance and fall to the soil, where they burrow and pupate. The adults emerge from the soil and the cycle starts all over again. In warmer climates, there can be 10 generations produced per year. Since their introduction to the United States and Hawaii, no real progress has been made in chemical control methods, though non-chemical control is very effective with the beekeepers I've spoken with in Hawaii. Beetle traps can be purchased, which encourage the adults to hide inside, where they are then trapped by vegetable oil. Alternatively, using brawny microfiber paper towels is also effective. The adult beetles get caught up in the fibers, and the honeybees entomb them with propolis. All you have to do then is remove the whole thing periodically and replace as necessary. Bet you didn't think that moths could be pests of anything other than tomatoes, did you? Well, the aptly named wax moth is the most serious insect pest of unprotected comb in warm areas. As with the small hive beetle, the larval stage is the economically damaging one, feeding primarily on the actual wax comb, cast larval skins, pollen, and honey. This is also one reason why used beekeeping equipment must be stored properly in Honolulu County, as it propagates pest problems. In strong colonies, honeybees will remove the wax moth larvae before they grow large, thus keeping the pest at non-economically damaging background levels. As the larvae mature, they create silk tunnels in which to protect themselves and ultimately form a cocoon in areas they chew out for themselves inside the hive bodies. When a colony becomes weak or absconds, wax moths will do significant damage to the comb. Thus, if you see cocoons inside your hive, you have a really big problem with wax moths being the least of your worries. The best defense is therefore maintaining strong, healthy colonies. If you do find you have infested comb, shake off the bees and place the frame in a freezer for 24 hours before returning it to the colony. This will kill the moths, and the bees will take care of cleaning it off. Cane toads are voracious predators of insects and small animals. They were purposely introduced in 1932 to control sugarcane beetles and are now invasive on all major Hawaiian islands. Though I haven't yet witnessed it, they are considered the most serious amphibian pest of honeybees and will enter through the bottom to eat the bees and larvae at night. To prevent this, keep hives raised off the ground. Cane toads suck, but hey, at least we don't have bears. Hawaii does not currently provide diagnostic services for pathogens of honeybees. However, my Pollinator Ecology Lab is in charge of administering the National Honeybee Survey in the state of Hawaii. We collect samples from commercial apiaries across the state, which are shipped back to Maryland, where the project coordinators run various tests for pathogens, conduct varroa mite counts, and sample for the presence of potentially invasive honeybee pests. Hawaii's participation in this nationwide survey is particularly important because we float between the mainland US and Asia and could intercept Tropolaelaps mites and other invasive pests before they make it to the mainland. Our honeybee industry, particularly the queen breeders, are essential for North American beekeepers. We can also provide unique data since we still have varroa-free islands. The data from this survey is all publicly available and can be accessed from the website listed here. You can view reports by year for specific states and see how their pest and pathogen loads compare to the national average. So, who's overwhelmed with information? Don't worry, you can rewatch this video as many times as you need to before taking Quiz 7 while the information is still fresh in your mind to make sure you get 70% or greater for credit for the Hawaii Master Gardener's Advanced Training. Stay tuned for the next video, which will be part 2 of pollinator declines, this time focusing on nutrition.